right, so far we talked about 4 ways that things can move in a cell. What's the most basic way? Diffusion, just simple diffusion. Things move from high to low concentration. They move passively. What kinds of things can diffuse through the cell membrane with very few problems?
Non-charged items, non-charged molecules, and small.
Yeah, non-charged and small. Those are the things that diffusion usually happens with. Non-charged and small things. They go straight through the main part of the membrane, which is the phospholipid part. We also talked about osmosis, which is essentially just the diffusion of what? Water. Eyes and ears officially. A little late for that homework. Put that away. Osmosis moves water from where there's a high concentration of water to where there's a low concentration of water. But we said before, a lot of times we don't talk about osmosis in terms of the water. We talk about it in terms of the things that are dissolved in the water, which are called what? Solutes. For example, today we're going to do a lab where we're going to have water in a cup. And we're going to put in one of these little dialysis bags, which we'll look at more in a second. And in that dialysis bag, we're going to have a sugar solution. Let's say in here we have a sugar solution. Okay. So the sugar solution has water in it, but it also has sugar in it. In the cup, there's just water. What should the water do? When we put the bag into the cup, is the water going to go into the bag, or is stuff going to come out of the bag? Water's going to go in. Because water always goes from where there's a higher water concentration to where there's a lower water concentration. Or from where there's lower concentration of what? Solute to where there's a higher concentration of solute. That's assuming that the sugar is too big to do what? Get out through the holes in the bag. And it is going to be. We're going to use a sugar that we've intentionally used to be too big to get in and out through those holes. All right, the third way of cell transport that we're going to talk about, and we talked about a little bit yesterday, Josh, put that away, please, is facilitated diffusion. We said facilitate means to give a little what? What's the word facilitate mean? Give a little help to something. So it's diffusion with a little help. This is the way that the big molecules and the charged things get in and out of cells, passively. The movement of from high concentration to low concentration through those little protein channels, those little tunnels in your cells. Again, this is mostly how ionic things, charged things, and big things move passively in and out of a cell. Remember, passive means what?
Use no energy.
Oh, okay. Well, let's be a little more specific.
It uses none of the. It uses heat energy.
Right.
Like other
It means the cell's not using its own ATP to do it.
Yes.
Okay? It still has to have energy because it's moving, but the energy to make diffusion and osmosis and facilitated diffusion is coming from where? The heat in the environment, thermal energy in the environment, not the ATP of the cell. So one other thing about facilitated diffusion that you should know is that these proteins that these things are diffusing through are really specific. There's certain channels just for like chlorine ions, Cl minuses. There's other channels just for sodium. There's other channels just for specific amino acids. But again, facilitated diffusion, diffusion with a little help. It's the movement of a substance from high to low concentration through proteins. It's a passive process. It's usually how ions and big things move in and out of cells. It's facilitated diffusion. Talked about that yesterday. The other thing that we talked a little bit about yesterday was active transport. Whenever we use the word active, that means what? It's the cell's using its own energy. It's using ATP to make it happen. You might think of active transport sort of as the opposite of facilitated diffusion. Active transport moves things against the gradient, which means instead of like usually we're moving from high to low, in this case, active transport moves things from where there's already not very much of something to where there already is a lot of that thing, from low to high. Sort of backwards from the way nature wants it to happen, right? This is important when your body needs to build up certain kinds of things in a certain place. We said it's really important in what part of your body, which is really important. Nervous system, like the building of sodium and potassium ions, electrolytes to make electricity. It's also really important in cell respiration. We'll talk about that a little bit later on. But it's like pumping a substance, pumping it against its gradient. ATP has to be broken down to make this happen. Cost energy for the cell. The one example that you should know of for right now is that guy right there. The sodium-potassium pump. Pump sodium and potassium ions. It's really important for your nerves and muscles. We'll talk more about that later. But there's another kind of a movement that we need to talk about today that we haven't talked about so far. He briefly mentioned it on the video we watched yesterday, but we didn't talk too much about it. This is going to be called endocytosis. And just break that word into its parts. What's endo mean? Inside. Cyto means cell. Inside the cell. This is when you're moving big materials into a cell. So far, the things we've talked about so far were just little atoms and molecules, right? Tiny things. Sometimes endocytosis is called bulk transport. Because we're moving big things, or big amounts of things. So it's endocytosis, moving big things into the cell. And we're going to break endocytosis into two types. I heard a lot of you giggling during the video yesterday because of that word right there. Phagocytosis. Get your giggles out of the way. PHAG means to eat. So it means cell eating. Okay? Cell eating. We're going to watch a video in a minute that's kind of cool. It's like a 30-second video. Leave this on the board? Or we should write it down? But phagocytosis is when a cell essentially engulfs a large solid object. For example, the video we're going to watch in a minute, an amoeba, little one-celled organism, you're going to watch it eat a living paramecium. Just kind of flows around it and pulls it in. We'll see it. We'll see a video of how it works. This is also how a white blood cell takes in like a bacteria or a virus to kill it. Phagocytosis. It takes in the whole thing. Once that material gets taken in, you got to break it down, right? You got to digest it. And that's where the lysosomes come in. Remember, those are those organelles with really powerful what?
Digestive enzymes.
Digestive enzymes. And they're going to digest that organism inside the cell. That's got its own little name. This was on the last test. What's going to happen after phagocytosis is intracellular digestion. What's intra mean? Inside. So something's going to get digested inside the cell. Intracellular digestion. Lysosomes make that happen. Again, we'll watch a video of one cell engulfing something large and solid and then breaking it down. We'll watch that video in a second. There's another type of bulk transport or endocytosis called pinocytosis. This means cell eating. What do you think this means? Cell drinking. This is when a cell takes in large amounts of dissolved solids. Take in large amounts of dissolved materials. And I'll show you pictures of how both work in a second. Sometimes those little protein receptors on the cell are involved in this process. We're not going to get too deep into this right now, though. So again, phagocytosis, cells are engulfing solid objects, and then usually they take them in, and they use the lysosomes to break them down, intracellularly digest them. Pinocytosis is when a cell takes in large amounts of dissolved substances, solids. And its receptors may or may not be involved in the process. What's the opposite of endocytosis? Something we already exocytosis. So endo is bringing in large amounts of things, exo is sending out large amounts of things. Those are both types of bulk transport, moving big amounts of things. We okay on that? You got what you need, Mark? Okay. So, now notice there's a typo on this page. This came out of the textbook, but this is supposed to be a G right here. It's not phagocytosis, it's phagocytosis. Here's a cell engulfing a red blood cell. So this cell membrane, is the membrane hard or not? Not. Okay? It's flexible. It's like liquid, right? So this membrane is going to move around the cell. It's going to surround it. It's going to start to pinch in on it. And eventually this side of the membrane is going to bump into that side. What happens when oil touches oil? It fuses, right? It becomes one thing. So that's what's going to happen here. Because this is made out of oil, right? It's made out of phospholipids, which are like vegetable oil. When this and this bump, they fuse together, and essentially they just bagged up that little cell. Now it's in a bag. What do we call that little bag? A vesicle or a vacuole. And then the lysosomes, let's say this is a lysosome over here, it's going to move over here. And what's it going to dump into that bag? What do lysosomes do? It's going to dump in those enzymes, and essentially that thing's going to get digested right inside that little bag. Broken down. Intracellular digestion. What now? Maybe it's an old red blood cell that's just not working anymore, and you need to reuse the pieces. Anybody know what organ in your body does that? Gets rid of old red blood cells?
The liver?
Kidneys.
The organ. Your spleen. That's why if you damage your spleen, you can bleed to death real fast because there's lots of blood going to it. But its job is to get rid of and recycle. One of its jobs is to get rid of and lower abdomen. If anybody ever had mono? Mono? The kissing disease. One of the things that happens in mono is that it infects your spleen, and your spleen sort of blows up almost like a balloon. And it's really dangerous because if when you have mono really bad, if you took a lick to the abdomen, it could cause your spleen to rupture and you bleed to death real fast. Yeah.
When I had it, my doctor said that I was super close to dying because it almost ruptured.
Yep.
And
Very
but
dangerous.
I didn't get mine from kissing on people. I got mine from drinking after people.
Sure.
I swear. I swear.
I would, yeah.
Is that what happened to them little boys who went to heaven and came back?
Oh, that heaven is real? I don't remember what happened. I read the book, but I don't remember what happened.
He said it was a
Is it dated? Yeah. What's up? So was it caused by mono? No?
It's called strep. Strep throat, and I got
So did your spleen swell up?
it swelled up through my fingers, and then I couldn't get down, and I had to stay in the hospital for a month.
That's not good. Good.
That's what people said.
The mumps is a virus infection. So I think you can catch it from the air. Don't quote me on that. But being around somebody who has it. But it causes salivary gland and stuff in that. So. All right. So one more picture here. This is a picture of pinocytosis. This is cell drinking. Remember, this is taken in dissolved solutes. Works pretty much just like phagocytosis. The cell membrane surrounds those dissolved materials, pinches inward, closes them off in a vacuole, and then it'll use them. Sometimes, like I said, it's triggered by little receptors on the cell membrane, sometimes not. All right, let's pause the video there for a second. Greener.